Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. In this video I am going to discuss the ultrastructure of renal corpuscle. Now what is renal corpuscle? Renal corpuscle is the blood filtering unit of nephron in which ultrafiltration occurs. It consists of Bowman's capsule plus glomerulus plus mesangial. Now this is the structure. This is the Bowman's capsule, this is glomerulus, and this is the DCT. Now this is glomerulus and this is the lining of capillary, that is endothelial lining of capillary. In between the endothelial cells there are some spaces. These spaces are known as fenestrations. The diameter of these pores or fenestrations is between 50 to 100 nanometer. It's important that the endothelial cells are negatively charged, so these do not allow proteins to pass. Then this layer is the visceral layer of Bowman's capsule, and it is made up of foot processes of podocytes. These are podocytes. In between these two layers, that is endothelial lining of capillary and the visceral layer of Bowman's capsule, there is basement membrane. This basement membrane is also negatively charged. Because of presence of negatively charged proteins like collagen. So this will also not allow the negatively charged proteins to pass through. Then these spaces in between the foot plates of or foot processes of podocytes are termed as filtration slits. Important thing is that these filtration slits are covered by filtration slit diaphragm, which is having nephrin molecules which are negatively charged. And these nephrin molecules restrict the proteins, the anions, and the high molecular weight substances, while these allow cations and low molecular weight substances. The filtration slits have diameter of about 20 nanometer. Now these all structures, that is fenestrations in between the endothelial lining of capillaries, basement membrane and the visceral layer of Bowman's capsule having the filtration slits, these are covered by a filtration slit diaphragm also. All these structures are combinedly forming filtration barrier. So we can say that the renal corpuscle is made up of filtration barrier plus mesangium. Now next part is mesangium. This is afferent arteriole. This is efferent arteriole. This is the capillary bed, that is glomerulus, and there are some cells in between the afferent, efferent and the DCT. These cells are outside the glomerulus. So these are known as extraglomerular mesangial cells. These are also known as Lassus cells or Burmacti cells. These are intraglomerular mesangial cells, and the spaces in between the mesangial cells is known as mesangial matrix. And these three, that is matrix and the two types of cells, are collectively termed as mesangium. Then what is the function of mesangial cells? Function of mesangial cells. First, phagocytic. These are phagocytic in action because these are derived from monocyte. So these are monocyte derived. So these are also the part of mononuclear phagocytic system. Second is, these give structural support to glomerulus. Next, these secrete mesangial matrix, cytokines and even prostaglandins. Then these also secrete some erythropoietin and some renin. Another important function is that contraction of mesangial cells alter the glomerular capillary bed area, thus alter GFR. So GFR is also altered. Next, it is also having role in immune complex disease of glomerular origin. So this is about the renal corpuscle and its ultrastructure. So hit the like button, share and subscribe our channel to get the latest updates and notifications.